Now we're going to talk about the nervous system. And to start with, we need to talk about the elements of the nervous system. The neurons are the real functional units of the nervous system, but neuroglia are important also as support cells. We're mostly going to talk about neurons. So the basic functional units of the nervous system, what makes it work, are neurons. And neurons are cells that are specialized to conduct impulses. We used to think there was no mitosis at all after birth of neurons. It turns out there is, but it's very limited, although that, again, that does mean that we might be able to tap into that and use it to replace neurons when they've been damaged, like because of a stroke or some kind of brain injury. But of course, we can't do that yet. Neuroglia are the support cells for the neurons. The neurons are so specialized that they can't really do all the functions that they need to do to stay alive. So the neuroglia are the cells that are doing those support functions. And there are a lot more neuroglia than neurons. It's about 5 times as many. And you can have limited mitosis in neuroglia and much more limited in neurons. So this is a basic drawing of a neuron. The cell body is the large part of the cell back here. It's where the nucleus is. Most of the cytoplasm is there. CNS is an abbreviation for central nervous system. So in the central nervous system, the neuron cell bodies are clustered together in groups called nuclei. And this is a problem, of course, because we start running out of terms. So this is not the same as the nucleus of an atom. It's not the same as the nucleus of a cell. It's a cluster of nerve cell bodies. PNS is an abbreviation for peripheral nervous system. And in the peripheral nervous system, the places where the cell bodies of neurons are clustered are called ganglia. Dendrites are not very well shown in this drawing, but the dendrites are the usually relatively short branches off the nerve cell body, and you can see they're all kind of fading off into nothing here. But they receive stimuli and then conduct that stimulus in the direction of the nerve cell body, so back towards the nerve cell body. The axon is the single long extension from the neuron. It can be, millimeters long or more than a meter long in the peripheral nervous system, which, even a millimeter long is long for a cell. There's tons of mitochondria in the axon because there's a whole bunch of membrane pumps that are basically burning ATP that are pumping ions across the cell membrane, so you need to be able to generate a lot of ATP. And this conducts the nerve impulse away from the nerve cell body. So neurons have a direction. Dendrites conduct the impulse towards the nerve cell body, and then once it reaches the nerve cell body, the impulse moves down the axon away from the nerve cell body. So this show is showing a myelinated neuron. Myelin is a lipoprotein. It's basically white. They're showing it kind of like a hot dog color here, which is not right. It's pretty white. Neurons can either be myelinated or unmyelinated. This one is a myelinated neuron, and you can see you've got these segments of myelin and then a gap. The gap is called the node of Renoir, and then another myelinated section, and then another node of Renoir, and it'll be like that going all the way down the axon in a myelinated neuron. Unmyelinated neurons just basically have a naked axon running down. So there's white matter and gray matter in the central nervous system. The white matter is where you've got myelinated neurons, and really the part of the neuron that's myelinated is the axon. So white matter is where you've got myelinated axons. And you can see in this diagram the way that myelin sheath works. It's actually a modified cell called a Schwann cell that produces the myelin, and it wraps around the axon. And so each segment where it's myelinated is one Schwann cell that's wrapped around the axon at that point. Unmyelinated neurons and nerve cell bodies are never myelinated, so wherever you've got a cluster of nerve cell bodies, it literally looks gray. Remember, myelin is pretty white, so where you've got a bunch of myelin, it's going to look white. Where you don't have myelin, the neurons look gray, so we call that gray matter. We can classify neurons based on how they function, what they do, and there are three different major categories of neurons. Sensory neurons, or another name for it is afferent, and I never like that term, but sensory neurons have impulses that originate in some kind of sense receptor and the impulse then moves towards the central nervous system. It's taking sensory information into the central nervous system. And then motor neurons, or another word for it is efferent neurons. Motor neurons have impulses that originate in the central nervous system and then move outward, usually to a muscle or to a gland, to make something happen outside the central nervous system. Within the central nervous system, you've got association neurons, or sometimes they call them interneurons, and those are the neurons that are connecting the sensory neurons with the motor neurons. So basically all the functions of your brain are being done for the most part by association neurons. The only functions that aren't are the sensory information just that's coming in as it's coming in, and the motor information just as it's leaving the brain. Everything else is association neurons. So the way that a neuron works, of course, is important, and it's been studied quite a bit. The nerve impulse is an action potential. So we're going to walk through how an action potential works, and I will have a couple of videos posted that also show what's happening, because again, it's a dynamic process, so it's often easier to see it happening than to just see static pictures of it. So when an action potential starts, you're starting with a neuron that's in the resting state. There are no membrane channels open. The membrane potential is negative inside the neuron and it's positive outside the neuron, and you can directly measure this. It's done all the time. Then once the action potential starts, the first part of the action potential is the depolarization phase, and when that happens, there are sodium gates that open that allow sodium ions to flood into the cell. Now, think about what that means. We just said that the inside of the neuron was negative. Now we're letting a bunch of positive ions flood into it. There are also specific gated channels that will only let potassium ions through, and there's a high concentration of potassium inside the neuron, but those channels stay closed. So positive sodium is flooding in, there's already some positive potassium in there, and because of all those positive charges flooding in, the neuron, which when it's resting it's negative, now goes to a positive membrane potential inside. Then the neuron starts to repolarize, and there are some gates that close on those sodium channels, so sodium stops flowing in. Potassium gates open, and there's a high concentration of positive potassium ions inside the neuron. And so when those gates open, those positive potassium ions start rapidly diffusing out of the cell, and of course, because they're positive, now we're moving a bunch of positive charges back to the outside of the cell, so the neuron is starting to repolarize the way, getting close to where it is in the resting state. And then the last step is the undershoot phase. The sodium channels are closed, the potassium channels are open, still open. The membrane becomes hyperpolarized, which means that it's more negative than it is when it's at the resting potential. And then after you get to the undershoot phase, you get back to the resting state. So let's take a look at a diagram that actually shows all of this. What you're looking at down here is the axon. This is inside the axon, this is outside the cell, outside the axon. And we don't usually worry too much about the negative ions, because it's really the flow of positive ions that's determining what's going on. But when we start with a cell that's in its resting state, we have a high concentration of sodium ions outside the cell and a low concentration of potassium ions. Inside the cell, it's the opposite. We've got a low concentration of sodium ions and a high concentration of potassium ions. And that concentration difference is being maintained by a membrane pump, and you can see it represented over here. So it's pumping out sodium ions and pumping in potassium ions, and of course it's burning ATP to do that. Now sodium will diffuse across the cell membrane. Potassium actually can diffuse out even a little bit faster. But so if the membrane pumps quit working, those concentration differences would very rapidly even out. They are maintained by the constant use of ATP to keep those pumps pumping the ions against the concentration gradient. So here are the 4 steps again, and the diagram in the middle is showing you the membrane potential, the voltage of the neuron as it changes. So we'll start at the resting state, which is at the bottom left-hand side of this slide. So you've got sodium channels and potassium channels, and these channels have gates on them. So there are protein channels that are specific. So the potassium channels will basically just let potassium through, and the sodium channels will basically just let sodium through. But there is a gate on them. In fact, there's 2 gates on the sodium channel that can stop the flow of ions through the membrane. And we already talked about how the membrane itself is a bit leaky, and you've got membrane pumps that are working. They're not shown here, but they're working in the background all the time to try to keep those ion concentrations where they're supposed to be. So this is the resting state down here. It's step 1, the neuron is not firing, and then a stimulus happens and the neuron starts to have an action potential. So we go into step 2 here, which is depolarization. When depolarization happens, the activation gate on the sodium channel opens, and that allows sodium to flood into the cell, and bringing their positive charges with them. So we're in the resting state, you see we're negative, but down here, as when the activation gates on the sodium channels open, positive charges flood in, so we go positive. And you can see up here, if you look at the potential there, we're positive. Then we go into the repolarization phase, and when that happens, the inactivation gate down here on the sodium channel closes, and that stops more sodium ions from flowing into the neuron. And the gate on the potassium channel opens, which allows potassium ions to follow their concentration gradient and diffuse out of the cell. So those 2 things basically happen at the same time, and when they happen, we stop the flow of positive ions into the neuron and allow the flow of positive ions out of the neuron. And so we start taking away those positive charges, and of course that pulls the membrane potential back down and makes it negative again. When we get to the undershoot phase, the gate on the potassium channel remains open. Both gates actually close on the sodium channel, and because the potassium gate is open, that channel is open, potassium ions keep diffusing out, and that pulls us more negative than the cell is going to be in its resting state. And then when we get out of the undershoot phase, the gate on the potassium channel closes, and the membrane pumps pump the ions back to the concentrations where they are in the resting state, and we enter the resting state again. So that's the form of the signal that the action potential creates. And one thing that's important about the action potential is that it's all or nothing. When an action potential happens, if there's enough of a stimulus to cause it, you're going to get an action potential. If the stimulus is just barely strong enough to cause it, you get the same action potential as when the stimulus is really strong. It doesn't matter. All the action potentials are the same. Now the other thing is, so once they start, they're going to go all the way through all these steps until you get back to the resting state. The other thing about the an action potential is that when an action potential is happening, you can't start another action potential. You can only have a neuron can only have an action potential when it's in the resting state. You can't start another one when it's in any of the other phases. It has to be back in the resting state for an action potential to happen. Now remember that the axons can be really long. I mean, they can be in a big person, they can be a meter long, and so that's incredibly. It's very thin, but it's still incredibly long for part of a cell. And so when the action potential actually is going down the axon, it propagates as a wave. So you notice that in this diagram here, we're in an early segment of the axon. The action potential has started, the sodium channels open up. The depolarization that's happening here triggers the channels in the next segment here of the axon to open up. So then they open up, and of course as they open up and allow sodium to flood in, we're moving into the next phase here, so the potassium is flowing out. And this depolarization that's happening here triggers the sodium channels in the next segment, and so they open up. So the action potential propagates as a wave down the axon. It doesn't happen all at once in the axon. It starts near the nerve cell body and propagates all the way down as a wave. Saltatory conduction is what happens in myelinated neurons. Saltation just means jumping. So when you've got a myelinated neuron, sometimes people say that the myelin is sort of like insulation on a wire, which is completely wrong. First of all, in a wire, you can't have little segments of insulation with bare wire between them. The whole wire has to be insulated except at the ends. But what a myelinated neuron does is it basically allows the action potential, instead of propagating it for every step down the neuron, it allows it to jump, or I should say down the axon, from one node of Renoir to the next. So you can get the action potential moving down a given length of axon much faster if it's myelinated than if it's unmyelinated. If it's unmyelinated, basically every bit of that axon has to go through an action potential. When it's myelinated, it's only really happening at the nodes of Renoir, and so it's skipping over the rest, and the action potential moves down very quickly. All right, so a nerve impulse has some properties. Nerve impulses only travel in one direction. The exception to that are neurons in Cnidaria, and remember, they barely have tissues, right? But they have two-way neurons, so the same neuron can be both a motor neuron and a sensory neuron, which is kind of an odd thing, hard to imagine how that works. But Cnidaria are the only exception. So in everything else that has neurons, each neuron can only send an action potential down in one direction, from the dendrites to the nerve cell body, and then down the axon to the axon terminals at the far end of the axon. So these nerve impulses are all or nothing. Like I said, there's no such thing as a strong nerve impulse or a strong action potential or a weak one. It either happens or it doesn't. All the action potentials are the same. The speed, though, is very different if you compare unmyelinated neurons and myelinated neurons, because again, if you have myelinated neurons, the action potential is basically just jumping from one node of Renoir to the next, and so it can go down much faster. So unmyelinated neurons conduct nerve impulses at about 0.5 meters a second. So that's pretty slow if you think about it. You know, somebody that's 2 meters tall, which is a bit taller than average, but there are certainly lots of people that are that tall. It would take an entire second for a nerve impulse to go from their brain down to their big toe, which is awfully slow. Myelinated neurons conduct impulses at around 130 meters per second. So it's not a little bit faster, it's massively faster. The synapse is the little gap between the axon terminal of one neuron and the next neuron. It might be the dendrite of a neuron, or it might be the nerve cell body, or it could be an effector cell like a muscle cell. And so there's a little gap there. There are some electrical synapses that exist. They are actually not very interesting. It's an electrical connection between neurons. So when the action potential hits the axon terminals, the charge differences that you're seeing when you get the action potential coming through actually trigger the channels in those, or trigger the gates in the channels on the next neuron and make it fire. It's very an electrical synapse is very fast. They aren't very common, and they're not very interesting because the synapses tend to be control points, and electrical synapses really aren't control points. Chemical synapses are the common ones. They're slower than electrical synapses. The connection between the two neurons, or between the neuron and the effector cell, is a chemical connection. So when the action potential reaches the axon terminals, a chemical called a neurotransmitter is released into this tiny gap, the synaptic gap or the synaptic cleft, and it diffuses across to the other cell and causes the the gates on the ion channels to open on the other cell. And it works basically as a control point. So here's a diagram of a synapse. When the action potential reaches the axon terminal, this little swollen bud here at the very end, it releases a neurotransmitter chemical into the synaptic cleft, which, because this is a tiny, tiny gap, it rapidly diffuses across and binds to ion channels on the next cell, the postsynaptic cell, and that makes those ion channels open. So, of course, you would have a problem if the channels on the postsynaptic cell stayed open. So the ion channels, the proteins that form the ion channels, will also break down the neurotransmitter. So you degrade it, and that way, once the nerve impulse comes, it doesn't just last forever and keep those channels open, because the neurotransmitter basically is temporary. It gets broken down pretty quickly in the synaptic cleft, and then to open them again, you need another nerve impulse to release some more neurotransmitter. And there are poisons that work by basically blocking the enzymatic action of those channel proteins so that you don't break down the neurotransmitter. And if you look at, well, I think a lot of the common bug sprays, if you look at the ingredients, it'll say it has, active ingredient is acetylcholinesterase inhibitor. Acetylcholine is a common neurotransmitter between neurons and muscle cells, and what that chemical does is it inhibits the action of the enzyme that breaks down that neurotransmitter, and so it basically kills insects because it makes their nervous systems just lock up, or their neuromuscular systems lock up. So this is a little bit more, slightly more realistic view of the connections between one neuron and another. So you notice that you've got, in this picture, you've got dozens of synapses, and they're color-coded in the drawing. The red ones are inhibitory synapses, so when one of the red synapses fires, that makes the neuron that you're looking at here less likely to fire. The green ones are excitatory synapses. When they fire, it makes this neuron more likely to fire. And so it's receiving signals all the time from lots and lots of other neurons, and basically whether or not this neuron is going to fire is going to depend on the sum total of all these signals that are reaching it. If the excitatory synapses can overwhelm the inhibitory ones, then this neuron is going to fire. And remember, this is only slightly more realistic. This is a really, really complicated network of connections, and we're just barely beginning to start to map them out and figure out how it works.